Okay, so this is chapter 7, part 2. And again, we're talking about cellular respiration. We have just finished glycolysis. And so let me take you to this slide. So remember, during glycolysis, we start with glucose. Glucose is a 6-carbon sugar. There's an energy investment phase and an energy payoff phase. And overall, at the end of glycolysis, we get 2 molecules of pyruvate. Those will be transported into the mitochondria. We also get 2 net ATP, and we get 2 NADH molecules. Remember, those NADH molecules are going to go all the way over here. They're going to go into the mitochondria and be used here in this last step, which we'll be getting to. So right now, we're talking about these 2 molecules of pyruvate. They're going to enter the mitochondria, and they're going to go through this first step in here called the formation of acetyl-CoA. Okay, so that's where we are. So again, pyruvate. Pyruvate is a 3-carbon molecule. 2 of them are produced in glycolysis. So again, we have to remember that this next step is going to take place for both molecules of pyruvate. So if oxygen is available, so only under aerobic conditions, pyruvate will be transported into the matrix of the mitochondria. And so again, here's our picture of where things are happening. Glycolysis happens in the cytoplasm. Pyruvate, the product of glycolysis, will move from the cytoplasm into the mitochondria, specifically in the very inside of the mitochondria, which is called the matrix. That's where this next step is going to take place. Okay, so this next step, which is the second step of cellular respiration, is going to be called the oxidation of pyruvate and then the formation of acetyl-CoA. Okay, so what happens here? So we say that pyruvate, both molecules, are going to undergo what we call oxidative decarboxylation. Okay, and we'll go over what those 2 terms mean in just a second. Again, this is going to occur in the mitochondrial matrix if we're talking about eukaryotic organisms. Okay, so what happens? So first off, a carboxyl group is removed as CO2. So there's one of our end products, carbon dioxide. So when we see the term decarboxylation, that means we're removing carbon, okay? And so this carbon dioxide that's pulled off is, again, a waste product. And that's in the main formula for cellular respiration, right? CO2 is on the right-hand side of that equation as a product. And so there's a molecule per pyruvate. So again, we have 2 CO2 that are going to come off. Okay, it's a waste product. It will eventually diffuse out of the cell. If we're talking about humans, it'll diffuse into our bloodstream and get carried to our lungs where we breathe it out. Okay. The second part of this, the 2-carbon fragment that we're left with, so remember, pyruvate is 3 carbons long. If we pull off CO2, we remove a carbon. So now we have we have a molecule that's only 2 carbons in length. Okay, so this 2-carbon fragment that we're left with is going to be what we say oxidized. And so you know that means electrons will be pulled off of it. And so what's formed? NADH, right? So electrons get pulled off and they get given to NAD to form NADH. Okay, and the last thing that happens is the 2-carbon fragment will get attached to what we call coenzyme A, and our final end product of this part of this reaction is going to be called acetyl-CoA. So again, it's a 2-carbon molecule attached to coenzyme A. I'll show you a picture of what that looks like in just a second. Here is an overall formula for this second step. So again, we have 2 pyruvates. They enter the mitochondria. They're going to be stripped of carbon dioxide. So here we go, 2 molecules of carbon dioxide, 1 per pyruvate gets removed. So that's that decarboxylation. Pyruvate will also get stripped of electrons, which are given to NAD+, forming NADH. So we say that NAD gets reduced. Pyruvate gets oxidized. And then this molecule here, where's my pointer? Coenzyme A, we need 2 of them, 1 for each pyruvate. And so in the end, we have 2 molecules of acetyl-CoA. Okay, those are our products. Okay, so here it is. So that you can see the structures. Again, I don't need you to memorize structures, but just so that you can visualize it. So again, this is pyruvate. It's 3 carbons long. Okay, and again, 2 of these are produced in glycolysis. Both are transported into the mitochondrial matrix. Okay, so what's going to happen here is this carboxyl group, CO2, gets pulled off. Okay, so there's the carbon dioxide coming off. Again, that's called a decarboxylation. We also remove some electrons, and so NAD gets oxidized to form NADH. So there's 2 of our products. And coenzyme A gets added. And so in the end, this is what we have. This is again called acetyl. So acetyl means 2 carbons, CoA. This molecule will now enter into the 3rd step in cellular respiration, which is going to be called the citric acid cycle. Okay, so again, some textbooks, some instructors don't actually teach this as a 2nd step. Our textbook does. This is the way that I learned it. Although I've had some instructors do it a different way. So I always think of glycolysis, then the formation of acetyl-CoA, then the citric acid cycle, and lastly is the electron transport chain and oxidative phosphorylation. So again, whether this is, step 2 or a transition step depends on what book you're reading, but we're going to call it an actual step. So this is step 2. So what happens next? So both of those molecules of acetyl-CoA that were just formed are going to now enter into the citric acid cycle, which still takes place in the mitochondrial matrix. So we stay in the inside of the mitochondria. So again, just to recap, from glycolysis, we end up with 2 ATP, 2 NADH, 2 molecules of pyruvate. The second step, which happens in the mitochondrial matrix, we end up with we start with 2 molecules of pyruvate. We end with 2 molecules of acetyl-CoA, 2 molecules of carbon dioxide, which are waste, and then 2 molecules of NADH. Remember, again, those NADH that are produced in both glycolysis and this 2nd step will end up going to the electron transport chain. Okay, notice that no ATP is made during this 2nd step. All right, we're going to enter into the 3rd step now, which again is the citric acid cycle. It takes place in the mitochondrial matrix. And we will have a net yield here of a bunch of things, but we will have 2 ATP that are formed. The ATP formed here in this 3rd step is formed just like it was during glycolysis by substrate level phosphorylation. So again, you want to make sure that you understand what that term means. Substrate level phosphorylation, you have a phosphorylated intermediate in which there's going to be an enzyme that's going to pull the phosphate off and give it to ADP, forming ATP. Okay, so it's a transfer of a phosphate. Very different from what we're going to see when we talk about the electron transport chain. Okay, so let me move my camera. So again, this is the 3rd step in cellular respiration. It is called the citric acid cycle. It is also known as the tricarboxylic acid or TCA cycle, as well as the Krebs cycle. So there are multiple names for this. I usually don't use TCA. I usually use either citric acid. Sometimes I call it the Krebs cycle. So you want to make sure that you know both of those terms. Okay, it's the same process. Again, the citric acid cycle is going to take place in the matrix of the mitochondria, so in that very inner part of the mitochondria. And remember that just like glycolysis and just like the last step, the formation of acetyl-CoA, there will always be enzymes catalyzing each step of these metabolic pathways. This pathway is made up of 8 steps. I don't expect you to memorize them, okay? But we'll go over briefly kind of the nutshell version, and then I'll show you the step-by-step version. So what happens is that acetyl-CoA, both of them, because there's 2 of them that are formed from 1 molecule of glucose. So that acetyl-CoA enters, and what's going to happen first is it's going to take the 2-carbon acetyl group and transfer it to a molecule called oxaloacetate. And the combination of oxaloacetate and that acetyl group forms a 6-carbon compound called citrate, and that gets the cycle going. the CoA, the coenzyme A that was part of acetyl-CoA is going to come off and it gets, kind of recycled. Okay, so that's how the citric acid cycle starts. Here it is. Okay, so again, kind of step by step. So here is the acetyl-CoA. Remember, 2 of them are produced from 1 molecule of glucose. This is going to enter the citric acid cycle. Coenzyme A comes off, and this 2-carbon fragment, which is called an acetyl group, will attach to oxaloacetate, which is a 4-carbon compound, forming citrate. So again, that's how the cycle starts. Citrate then undergoes some reactions where electrons are pulled off to form NADH. Carbon dioxide gets pulled off. We end up with a 5-carbon compound that again gets oxidized to form NADH and decarboxylated to form CO2. So again, we're pulling off electrons, we're pulling off CO2. We end up with a 4-carbon compound. There is going to be a transfer of a phosphate group. So again, substrate level phosphorylation happens right here to form ATP. We also reduce FAD to form FADH2. Remember, this is NADH's cousin. It's the same thing, it's just an electron shuttler. So here, more electrons were pulled off. Water gets added in, and again, electrons get pulled off again to form NADH, and we recycle our starting material, which again was oxaloacetate. So it's a cycle. We have to go back to the beginning, right? And so oxaloacetate is ready for the next round. Okay, so we've got reduction events, right? NADH and FADH2 are formed. Both of those molecules will take those electrons that they're pulling off, keep them in the high energy state, and bring them to the electron transport chain, right? They're electron shuttlers, which is equivalent to energy shuttling. Okay, and then carbon dioxide is stripped off. So by the end of the citric acid cycle, we say that glucose has been completely oxidized. Okay, it's been completely broken down. All of the energy that is capable of being harnessed has been pulled out of that molecule. Okay, so here it is in words. Oh, pardon my dog. Okay, so again, citrate. So acetyl-CoA, the acetyl group is going to combine with oxaloacetate, forming citrate. Citrate then goes through this series of chemical transformation, losing carbon dioxide, forming ATP, forming NADH, and forming FADH2. Those are the products. And again, in words here, by the end of the cycle, glucose, which was our starting material, is completely oxidized. All of the CO2, all of the carbon dioxide has been generated. Okay? Let's go to the next slide. And here is some more words for you, just so that you know all of the totals. So I did this. This is the same slide that we just saw, but I want you to know that for every glucose molecule, 2 acetyl-CoAs are produced. And so we have to think about 2 rounds of this cycle. And so in total, this cycle produces 6 molecules of NADH, right? So we got 1, 2, 3 times 2 is 6. So we got 6 molecules of NADH produced, 2 molecules of FADH2. So here's FADH2, we got to multiply that by 2. ATP, 1 per acetyl, so 2 total. And 4 carbon dioxides are produced, right? So here's 1 and 2 times 2 is 4. So by the end of this, all of the carbon dioxide has been pulled out. 4 molecules here and 2 molecules in the previous step. Okay, so the formation of acetyl-CoA also produced 2 molecules of CO2. So again, glucose is completely oxidized by the end of the citric acid cycle. I have to show you the nitty-gritty just because. Okay, so again, I don't expect you to memorize this, but I wanted you to see all 8 steps. And so if we start at the beginning, here's oxaloacetate. It's a 4-carbon molecule. It combines with the acetyl group to form the 6-carbon molecule called citrate. Citrate then becomes isocitrate, which is oxidized and decarboxylated. So here's NADH, here's CO2 that gets pulled off to form alpha-ketoglutarate. This is a 5-carbon molecule. Again, CO2 gets pulled off, and so do electrons. NADH is formed, and we form what's called succinyl-CoA. Here is our ATP formation by substrate level phosphorylation. We form succinate. Succinate, again, is dehydrogenated, right? So it is going to be oxidized to form FADH2. We get fumarate. Water gets added in. We end up with malate. Malate, again, gets oxidized. Here's our NADH. And we recycle our oxaloacetate. So again, 8 steps, 2 rounds per glucose molecule. Okay, so you want to make sure that you know the totals of stuff that's produced. Again, you don't need to memorize every single step of this pathway. Okay, so what happens next? So in part 3, we're going to talk about this last step, which is the electron transport chain and chemiosmosis, oxidative phosphorylation. We'll use all of those terms. And so again, this is where the bulk of the ATP is going to be made. How? So we have to, again, bring this home. So all of the NADH produced during glycolysis, 2 of them. All of the NADH made during the formation of acetyl-CoA, 2 of them. And all of the NADH and FADH2, so 6 NADH, 2 FADH2 from the citric acid cycle, all of those electron shuttling molecules are carrying those high energy electrons that they've stripped off of glucose along the way, and they're going to carry those electrons to this very last step. And it's in this last step that those electrons are going to slowly lose energy, and that energy is going to be used to make a lot of ATP. So that'll be in the next video.